I'm excited to welcome everybody to this new series on early career workshops in respiratory omics, co-sponsored by the section Genetics and Genomics and the Assembly on Allergy, Immunology, and Inflammation. So there's a lot of great resources for postdocs, career faculty members on their path to independence. But this workshop is really supposed to add to those other resources and really focus on navigating the world of respiratory omics. So today we're going to be talking about collaborations in omics. We're very lucky to have two giants in the field of respiratory omics here to guide our discussions, Dr. David Christiani and Dr. Mark Wurfel. If you would both introduce yourselves briefly, that'd be great. We could start with Dr. Christiani.
Thanks, Matt. So I'm Dave Christiani from Harvard Medical School and School of Public Health and Mass General. And I'm a physician, and also I do population studies, molecular epidemiology. Oh, I see one of my former trainees sign on, Ju Jiajong. Hi. Hi. And so I started on this path in the early 90s, interested in sort of toxicogenomics, pharmacogenomics, and genetic regulation of the effects of exposure to various toxic agents. And then that evolved into lung cancer work, and then later, even though I'm a pulmonologist, only later really in non-malignant related lung diseases, and that evolved into sort of multi-omic studies or transomic studies later. So good to see you all.
Great, Dr. Wurfel.
Yeah, Mark Wurfel. I'm a professor of medicine at University of Washington, based at Harborview Medical Center. So I'm a physician scientist. I attend in the ICUs here, and my research for many years is focused on translational approaches to understand the molecular risks, molecular and genetic risks for organ dysfunction and death in critical illness. I've mainly focused on sepsis and ARDS, more recently AKI, and I came at it from a PhD in immunology, so I definitely was more of a molecular and mechanism guy coming into it, and then I sort of transitioned into more of a content person in ARDS, and so I think of myself as more using genetics and genomics as a tool rather than being an expert in it. And I think that sort of may be relevant to this discussion, and part of the reason I was collaborating with David in the first place.
All right, great. Thank you very much. So I'm Matt Mall. I'm a pulmonologist, junior faculty member at Brigham and Women's Hospital and Harvard Medical School. We have three co-facilitators. If you guys could introduce yourselves, that'd be great. Rachel, would you like to start?
Hi, I'm Rachel Kelly. I'm also at the Channing Division of Network Medicine with Matt. I work primarily in metabolomics and integrative omics.
And Ian?
Hi, my name is Ian Koenigsberg, and I'm an instructor at the University of Colorado Medical Campus. I have a PhD in human genetics, and I'm interested in omics modeling and complex lung disease.
And Katie?
Hi, I'm Katie Allen. I'm a lead scientist in the Department of Host Microbe Interactions at St. Jude. I work with Paul Thomas, and we essentially use human cohorts and genetics, immunology, and infectious diseases to figure out aspects of host response to mostly flu, but also a lot of other lovely respiratory pathogens.
And so Katie's going to kind of start our discussion here with the roundtable discussion and a little introduction.
Yeah. So first we'll start out with just kind of a overview. So the main discussions in this workshop are going to be recorded and will be posted on the ATS website, as well as put out in the AJRCMB Lung Science podcast, which is a series that's aimed at delivering engaging interviews and commentaries with luminaries in pulmonary and critical care basic investigation. The breakout sessions that we're going to have a little bit later are not going to be recorded. If you do want to pose a question throughout any of the discussion sessions, just type it into the chat feature of the webinar, and we'll present it to the panelists. So first, we're going to start out with the roundtable discussion with questions for our panelists, and then we'll move into breakout sessions and then reconvene to discuss everything that we've already talked about. Okay, I'll start out with a question posed to our featured guests. How do you define collaboration in your own work?
Mark, you want to start that?
Okay. I mean, I guess I hadn't really thought of it in terms of a definition before, but I think maybe it dovetails with what I just said. So I think as you look at a question, a scientific clinical question, you think about what you contribute versus what someone else might contribute, and I think genomics is a place where there are certainly lots of people who have some knowledge of what genomics can give you in the way of answers and the basic approaches to doing that. But I think there's a big difference between having some dilettante knowledge and actual understanding of how to do it and what the limitations are. The flip side is, you know, the genomicist's understanding of what mechanism is and the implications of what you might find using a particular study design really kind of relies on the epidemiologist and molecular biologist to help inform. And so when you're thinking about collaborations, I think it's particularly in the genomic space, I think it's really helpful to have expertise in those realms. And I will say that having sat on an NIH study section for these sorts of studies, that was a critical thing that you look at when you're evaluating the likelihood that a study is going to be successful.
Yeah, I would agree. And that could dovetail into the next section you have, Katie, on the different types of collaboration. So the one, as Mark described, is a complementarity. People, it's the highest level of collaboration. It's scientific levels where people bring different expertise, but the whole is more than the sum of its parts. And so that's the ideal. The types of collaboration can be quite kind of broken down, the sort of driven by the sort of scientific questions, as Mark said, but needing additional resources, population resources, lab resources, or whatever, where it would be necessary and important collaboration, but maybe not necessarily driving all of as the first two or three individuals. Or usually you need a small core group to drive these things, you know, because we're all busy. And the more collaborators you have, the harder it is to get everybody together, even just to talk. So to get the drivers in the driver's seat to move it along, sometimes you do need, and now even the other types of collaboration that hopefully won't take too much time from people who are not in the driver's seat but willing to participate. So then you start to see the group form, and that can relate to some of your other questions about issues.
No, that's really true. You know, you've both spoken a little bit about the kind of, in a way, different types of collaboration, but one of the questions that I've always had dealing with the different types of collaborations is timing. And that's kind of, it's always hard because sometimes, you know, your data leads you in a certain path that you're not expecting. But when you have a big aim, let's say, that's going to involve genomics, do you like to involve your collaborators far in advance, like in the planning stages, despite the fact that, you know, it is difficult to have everyone come together, just like getting everyone on the same calendar, but knowing that that expertise could be really helpful in the beginning stages of a study? Or is it something that it's just like natural, and when you know you need someone, that's when you reach out to them?
Yeah, I think number two more than number one, as much as possible. Right, Mark, what do you think?
Yeah, I agree. I think it slightly depends on what stage the data are at. So, you know, I think that there are different approaches. Much of what junior investigators are going to do is going to be reusing existing data, right? Repurposing it to answer a new question. And I think that may start with you and then evolve into whatever content expertise you might need for that, other measurement expertise, epidemiologic expertise, whatever, as opposed to the sort of planned prospective study where I think, while you may come up with the initial inkling of what's going to be done, I think in the prospective study, I think it probably is helpful to bring everybody together a little bit quicker, just so that it keeps everything on track and efficient.
It's really valid. Okay. The last big question that I have, and again, people put questions in the chat box if you have any. What issues related to collaboration are unique to the omics field? And are there special considerations in collaborations involving different types of omics data?
David, you want to start?
Yeah, I think when you're now talking about transomics or multi-omics, we're talking about platforms where people spend whole careers developing that particular branch of this phylogenetic tree we're talking about. And you're trying to bring these together into the trunk and into getting something done. That could be quite challenging. And so if you're really starting to do not just omics, but again, multi-omics, there are different issues with each of the platforms, lots of technical issues. There are some statistical considerations, and all of those really have to be dealt with as you're starting to work together, and knowing particularly the limitations of each of those platforms. And it's not going to be all cured by statistical methodology if there are intrinsic problems, particularly generating the biologic data. And Mark's got a lot of experience with this because he's actually been situation in the lab having to grow cells and then a study say, well, we really need to know the function of this. So Mark, can you just like grow these things and then tell us what it does? So he can expand more on this, because that is a lot of work to get to that point. Not all the data is in the public realm yet.
Yeah, I mean, there are lots of things that are specific to multi-omics, but I think there's also some general problems. So I think that I'm sure everybody's aware, but the one of the big problems is just data sharing, data alignment, data use, all of those things that actually just kind of get in the way of collaboration for a multi-omics study. So I would say they're a bit more of an issue in multi-omics studies than sort of large epidemiologic studies. There's still data use issues, definitely transcend all of those kinds of studies, but I think it's something that we particularly struggle with, just because of the kind of big collaborative nature, and coming up with appropriate use agreements and collaborative agreements can be challenging. But I think David raised a really important issue around sort of the data generation step, and understanding data generation and all of the issues that come along with batch effects, location of measurement, all timing of measurement, all of those things can have huge effects on the analytic product, that if you don't understand up front, could give you a big fat result that's not valid. So I think that probably more than many other disciplines, it is an issue you really have to tackle up front.
Yeah, and I definitely think with omics, generally the size of the data that we're trying to share and collaborate on always causes issues. And I feel like in the omics field more than others, that's just a headache that we always have to deal with. That's more technical, but it is something that we've run into a good bit.
And I guess I would just there's I think in this group, we think a lot about the molecular aspects of maybe batch effects and measurement errors and things like that. But on the epidemiologic side, I mean, particularly when you're talking about in what I study and David studies in critical illness, the data stream, the clinical data streams, and how you collect those clinical data streams, the specific tools you're using as far as CRFs and such, also can have huge effects on the analytic product that really need to be addressed.
Yeah, there's excellent points, Mark, because there's a tendency to look at data that's there and then to analyze existing data without necessarily understanding all the limitations of some of the data. And some of the things Mark raised are very fundamental. They're not just technical, although we just talked about a number of technical issues. There's still study design issues, population issues that are very fundamental. It's almost like Epi 101. Some of the early studies, I mean, we were doing gels back then and stuff in cancer, but then I got into ARDS in the late 90s because I published a bunch of stuff in cancer, and my chief at MGH said, "Oh, the NHLBI likes what you're doing in cancer. Can we do some of that in critical care?" It actually wasn't that easy to move into the ICU to do this kind of stuff. But one of the things was study design. So it's, in a lot of the genetic studies, combining data from different populations, you never get a sense, at least early on, it's better now technically, but how many samples were rejected in the genotyping. And it gets even more complicated when you move to gene expression, some of the other platforms. They just tell you what they have. They don't tell you that maybe one site had a 20% rejection rate and have some very questionable issues about their design and about their sample quality. And another one had a 2%. You never saw that. It just basically, it was a geneticist issue. It wasn't because they weren't genetic epidemiologists like Ed Silverman. They were geneticists. They didn't really think about bias and confounding. They got data. You got a person and you have the data output. So what's the problem? We're just combining and then we'll adjust for covariates. And it's really some substantial epidemiologic issues, design issues about bias that can be introduced when you don't actually understand the origins of it, the structure of the population and the structure of the data. A lot of that now is being settled because of lots of data and lots of good techniques to deal with that. But there was a lot of kind of messy studies, I think, for the first decade or two on this.
And there is a question in the chat. I think we can we do want to move to breakout sessions soon, but we can address this. So from Zhu, as the many omics data were generated in the past years, what are the future directions of omics research in respiratory diseases, and how can we collaborate in this direction?
I'll start with that. That's a very broad question, Zizi, but I think what you're getting at is, now, are we building a big enough and hopefully publicly available database, things like the UK Biobank and others that have well-phenotyped chronic respiratory disease? Again, Mark and I have studied acute problems, but we really want good phenotypic data, something also that kind of relates to what I said before about study design, is well-phenotyped, well-characterized disease with genotype data and gene expression data, metabolomic or proteomic data. Where is that going? It really depends. It's going to be going at different rates in different diseases, right? Those are bigger public health impact, like asthma and COPD, likely going to be generating a lot more data as you're saying, in the near future. And those that are rare, it's going to take longer to get sample sizes large enough to start. One of the trends, I think, is also to look at shared genetic variants among respiratory disease. Because as the data grows, and we actually just have a paper that will be coming out in translational lung research with my colleagues from Nanjing. We looked at 5 respiratory diseases in relationship to lung cancer and shared genetic traits. And that's going to be exciting in the future, because you're going to start to not just do disease by disease, but we do like COPD, asthma, interstitial fibrosis, pneumonia. I mean, you can actually put a lot in there and start to look at some shared genetic traits with different outcomes. Not to let the data do the driving. You need to still have surely some good hypotheses about this, but I think that it's going to be some interesting trends, at least in the chronic disease realm, and separately kind of in the acute disease realm. I don't know, Mark, what do you think about that?
Yeah, I mean, I won't belabor it, but I think the two things that I see are that as we move towards doing a lot of our data collection electronically through the EHR, the critical thing is going to be, if it garbage in, garbage out, right? So being very careful about phenotyping, as David alluded to, is really going to be important. And I think as the databases increase in size, even some of the rare diseases are going to be addressable. like, ARDS isn't the most rare, but it is much rarer than asthma and COPD. And to address that, it's going to be important to have phenotypic measures that are actually aligned across different EHR databases. So that's going to be critical. And then, I think everybody knows that computing power, AI, machine learning, whatever you want to call it, is the next step. And so having the well phenotyped data sets combined with the multi-omic data sets will produce useful results as opposed to a lot of artifact.
All right, great. Well, welcome back, everyone. So we're going to summarize the discussions and get feedback from our panelists and extend the discussion a little. So group one was led by Katie, if you want to.
Aisha is going to be our spokeswoman.
Great.
Okay, so, can someone, I don't remember the exact wording of the question. Katie, can you read the question and then I'll summarize?
Yeah. So how do you identify collaborators in the omics world?
Sorry, I had a mouse issue. Okay, so we had a really great discussion group, and there were one of the first point, some of the easiest ways is to go to people that you know at your institutions. Next is to ask around, perhaps neighboring labs or even people within your lab. And even thinking of outside of your institution, you can ask people at conferences, meet new people at conferences, and that also using your connection. So maybe it's contacting editors at journals or using your connections at ATS through smaller groups such as the section on genetics and genomics. And that can help you identify and get connected with people that can help you get involved in groups or meet people that you would like to collaborate with. And so one example of a group that you might try to get involved with are these working groups for larger cohorts like TopMed that can, help you meet people, help people know what you're doing. And with all of that in mind, that highlighted, two things for us. One is the importance of networking and also institutional and society presentations, because it helps people learn what you're doing, what your interests are, in case you're someone that they would want to collaborate with. But then it also helps you learn what other people are doing and what expertise they have, so that you know who you might want to be collaborating with. And then the other is just that it's really helpful and nice to be able to collaborate with people that you like, that you can trust, and that you are able to rely on. It helps with the communication, problem solving, to be able to optimize working together and getting the best data.
That was perfect. I think we were going to see if anyone has any comments on this aspect of omics collaborations. Dr. Christiani or Dr. Wurfel, if you had any thoughts.
I guess all I would add, I mean, it sounds great and appropriate. I think the one thing, and it kind of came up a little bit in our discussion, I think it also depends on what stage you're at. So if you're very early and you're putting together a career development award versus putting together a large R1 or independent funding, that the goal of the career development, it definitely includes a lot of training aspects. And so the sort of person you might select to be a collaborator, mentor for that kind of role is obviously not necessarily different, but could be different from the kind of person that you would select for an R1 or something like that. And getting, because of the fact that you'll need access and you'll need somebody who is, interested in actually teaching you as opposed to just giving you access to the data, I think just is an important consideration.
Yeah, I agree. I think these are all good points.
Okay, should we go on to group 2? Rachel, your group?
Our spokesperson is Nicole.
Hi, so our question was how to establish new collaborations, and we kind of broke this up into two kind of bigger questions. So Dr. Christiani kind of offered one of the good ways to start thinking about new collaborations is to identify areas where you're deficient in, whether that's like a disease area or a new omics area, for example. And when we talked about it as a group, we talked about possibilities for that being things like involvement in consortia or professional groups that can help you identify potential collaborators. Obviously, some of the things that the last group touched on for other points like networking and your already existing circle. And then we kind of moved on to also speak about how to form collaborations when you want to like have access to things like new data sets in the context of being a junior researcher. So what challenges are kind of faced for people like us at like postdoc or early faculty. So using mentors or people you've collaborated with during your postdoc or PhD time or clinical training. But I think that Rachel pointed out a really important point that sometimes this creates like a small cluster or groups of omics investigators. So it's good to also consider how we can branch out. So things like willingness to be involved through those types of networking opportunities can help. And I think actually, Dr. Christiani, what you were saying, I might have gotten kicked out of the group before I fully got that. So I'd be happy to hear if you want to expand on that.
Yeah, well, I just think conferences are good. We're coming out of this dark age of COVID, but I think it's important to actually network at conferences. It's quite good. And the other thing that just came to mind is, it depends as you approach consortia, not all of them are the same. They may have certain principles, guiding principles. They all should have written guiding principles of operation with steering committees that are good. Some are more chaotic than others. Not everyone is as tight as like UK Biobank or something like that. What I've noticed, for example, so I sit on the board of, and Mark and I were on an ARDS consortium a long time ago, ISPAR, and then I sit on International Lung Cancer Consortium, and we now have about 100,000 cases in control. So we get a lot of proposals. But the way our approach is, because you're concerned as a junior person, do you really have a unique idea, or is it like the same idea that a lot of other people are working on, so you're going to get shot down right in the beginning? That's a big problem. That's a problem with some of the smaller cohorts that are not necessarily the huge consortia. For example, the Harvard cohorts. If you come up with an idea that someone else is working on, you're not going to be able to actually do it. But a big consortium like ILCO, what we tend to do on the steering committee is, this is a good promising junior faculty member. The idea overlaps with a group that's already begun working on it, but we try to get them to work together, try to bring them into the fold rather than just say you're out. And that's generally worked pretty well. It does expand the number of people on the steering committee that, it's more than just a few sometimes, but it often helps to use that approach to kind of draw junior people in. And there's usually plenty of work to do. I'm surprised at how much comes out of UK Biobank. We certainly got a lot out of it, where there's thousands of protocols. I kept thinking when we approach work in that is, well, someone must have thought of this before. Otherwise, but it's amazing how much can be done out there.
Yeah, and I would echo that. You ideally want. I mean, it's a very good thing for a junior investigator to be part of a network of existing data collection approaches, et cetera. I mean, you just don't have the resources when you're starting out to be able to do things on your own. So that's really important, and it's a great way to get connected with people. But I think what David says is critical, that you want to understand what the processes for new ancillary study approvals are, understanding that they are actually disseminated across all the investigators. They have some kind of functioning system to disseminate it across all the investigators, so that you don't end up with either being rejected out of hand. That, as David said, really the best way to approach it is to try and bring everybody up and develop inter-junior people collaborations that will work. But sort of the worst case scenario is you progress along, and then all of a sudden somebody comes out of the woodwork and says, no, that's my project, and you've wasted a lot of effort. So I think it's really important to understand who, what networks you're collaborating with, and you should use your mentors and, local expertise to try and understand which ones those are.
Is that good for group 2? Those are really amazing things to note. Okay, group 3, Ian.
Yeah, so our question was, once you have established contact with a potential collaborator, what are critical questions and issues to address? I am the spokesperson for the group. We were a fairly small group, but I think that was great in that we had a lot of time for a relatively early career faculty member to ask Dr. Wurfel sort of pointed questions about launching a new collaboration and really, I think, major wrenches to avoid. And I think we covered a lot of important points there, some of which we've already sort of covered, but really hammering down this early on, establish what the approval procedures are and make sure you're sort of following them. Yeah, I think a lot of things sort of revolve around this idea of making sure you play ball early, make sure you know what you need to do as an investigator, especially considering things like secondary approvals in these contexts can often take months and months to get through. One thing we talked about that I can echo is like a very effective approach is leaning on a senior, more established person, especially when you're entering a new collaboration or a new consortium. If there's someone who's gone through a lot of these processes before, such that you don't really have to reinvent the wheel for yourself. And again, establishing a lot of things early on and upfront, such as workflow and authorship, and a communication plan as well. Dr. Wurfel, is there anything you'd like to add from our conversation?
No, I think one thing that came up was the issue of to what extent you involve the collaborators, if they're just data access people versus actual content experts versus mentors, right? And so I think you don't necessarily have to have everybody maximally involved. It really should just be appropriate according to what stage and what kind of award you're searching for. And so if you're an early stage, you want, kind of apropos what I was saying before, you want somebody who actually has some track record of bringing up junior people, just because that's going to be most helpful to you. Whereas as you move along, really what you might need is, true content experts and leaders in the field, such that you get the best data and access.
Thanks so much. Dr. Christiani, is there anything you'd like to add?
I think I agree with all that. I think because you can break this down into willingness, the scientific enthusiasm, that could be very good. Let's do it. And you're going to be brought on board and get access. And then you sort of have these other issues that you can almost put into an administrative category, like the data use agreements, which can take a long time because of the institutional issues. Generally, the IRB issues are not big, but those are things you should pursue after you get the sign-on to do scientific collaboration, and then get moving on them because they could take a long time. Things like authorship, that should be part of the collaboration discussion. And the best of your ability, it really should be written up in a MOU, or if there's an application to a consortium to use their data, they usually have a form like that, with a proposed authorship group. That the order can change as projects evolve. Things can change. You run into problems and you need to bring on some additional help and analysis. And it's perfectly fair to say that person who's going to bail everybody out gets elevated in the ranking of authors. So that should just all be spelled out in the beginning, especially with junior people involved. It's really critical to have your senior mentors and the senior person's colleague who you were negotiating with in the other sites to be on board, so that those other sites, there's no misunderstanding between the senior person and the junior people in that group. So the best way to do it is essentially like a good MOU or template, a proposal template. And again, realizing that things could change some, hopefully not dramatically in terms of who's the first or co-first authors, but it could well be, and I've seen situations where we run into trouble and you really need another kind of data scientist on the project to help, and that person puts a lot into it. They should be recognized as such. And so, once the relationship is sound enough, these things can be negotiated. What you don't want to have is a weak link, something that goes on and the person proposing it was not committed enough to the project, and the whole damn thing starts to get shaky and everyone's not too happy to carry the ball. But we're assuming that the junior investigator here is enthusiastic enough to carry it through to the end. And this funding and stuff, you got to talk to your mentors about that.
These were all incredible points. I wanted to ask a question that I had missed from before, and I'm sorry, Eric, for not seeing this, but it asks for early career individuals, when do you recommend is a good time to reach out to others to start collaboration? Is it to use it as validation, data sets for your primary work, or to use for training grant proposals, etc.? So timing and types of.
I think our theme is reach out early and often, because the scientific reasons to collaborate with somebody are necessary, but there are all the other issues that we've been talking about that can actually torpedo even the best scientific collaboration. So I think really establishing that the scientific reason to collaborate is there and maybe mentorship reasons are there, but the sooner you can get to the logistic issues, the better.
Great. There's another question in the chat. So from Amali, so when using publicly available data sets, how should we be thinking about placing all these new analyses in context so that each analysis has an impact in future analysis? And in this vein, how do we increase visibility of analyses as not all publications are equally cited / referenced, especially if the omics analysis reveals trends not particularly of relevance to that scientific question or objective of that project? I assume that means the initial cohort project. So, how really is a question about how to leverage publicly available data sets for other types of projects. Is that right, Amali? And their intended use?
Yes, thank you, Matt.
So I think I understand the question. I mean, most of what we're talking about are going to be what Mark referred to as secondary analyses and ancillary analyses for. So if you're kind of assuming that you're not going to be running into the main purpose of a study or a group, a consortium, that most of the main effects probably would be already published, I would think, at this point. But maybe I'm wrong, unless I'm misunderstanding. I don't know, Mark, do you have a different question?
Yeah, I think I understand the question, but I think David said what I would have said, that you're going to have the primary question, and that, I guess, one thing, and I've seen this happen in other consortiums, that junior people don't understand what the primary question was, and so they start proposing stuff that's directly conflicting with the primary aims of whatever project. So I think understanding that up front is important, and then how you leverage the data into something new and impactful. I mean, that's just strategy. Talking it over with your mentor, that's understanding where the knowledge gaps are and what's going to be impactful. I think there's no secret sauce for that.
Now, do you think that approaching, if you have an idea, leveraging publicly available data that's off target from the original purpose, approaching the consortium or the group and saying, look, here's my idea, but we want you all on board, and actually forming a collaboration?
Yeah. Yeah, that's a very good point, because I've got requests from our own cohorts, not necessarily just from the consortium, is, can we just have all your data? like everything. It's for lots of reasons that's not possible. So those are sort of naive questions that come from a lot of places, and it's obviously probably not well thought out with a mentor. But I agree with you, Matt and Mark. What's your comments on this? Sorry.
All right, great. Well, so we've covered a lot of stuff today, right? I mean, we talked about sort of the nature of collaboration, timing of collaborations, doing, like you said, doing it, reaching out early and often, thinking about the technical aspects of things, but also, what role is a collaborator going to play? Are they going to be just a data access person? Are they going to be a mentor? Are they going to be a content expert? We talked about many approaches. I thought there was a really nice discussion from the groups about how to identify collaborators, how to establish collaborations, and how to really address issues of really laying out a plan and protecting junior investigators early on by having the details. So I really appreciate the discussion from everyone so far. For our panelists, do you have any other kind of words of wisdom or thoughts or comments to add? It's okay if you don't.
Just don't get discouraged. You're gonna, just as if you're entering Mark's lab and growing cells, you're gonna fail with experiments a lot in the beginning. So why should it be any different when we're doing population work? Don't expect to be a success for everything you do. Don't be a Gen Z or whatever the hell it is that my kids do. That seems unrealistic. You're gonna have to fail. The best success in many ways is failure, at least in the beginning. Not to quote Bob Dylan, the best success is failure, and failure is no success at all. We don't want to go that second route, but I think don't be afraid to make some mistakes along the way. Again, as long as you have good mentorship, good collaborators, then you correct each other. Manage up. Make sure your mentor is on top of this stuff.
Yeah, that's a good point. Very good point. And I think I would just say that hopefully you don't fail, but I mean it happens, and you need to just move on. But I think the biggest problem that I see is that people have overly expansive ideas about what these kinds of initial collaborations will produce. And really, I think at the early stage, you should think of it as an education and process and analysis and getting you to where at the next step you're going to be being able to address the really, really critical questions. So be mindful of expectations.
All right, great. So this is great. So this brings us to the end of our SCG Early Careers workshop on collaboration in omics. And again, thank you to our panelists, Dr. Christiani, Dr. Wurfel, to our co-facilitators, Katie Allen and Ian Koenigsberg and Rachel Kelly. Definitely the staff who helped organize this, Miriam and Erica. And definitely everyone who came here and participated today. Remember that the workshop was recorded, minus the breakout sessions. And thanks to Eric Morrill, this should be available via the Red Journal Lung Science podcast. If you enjoyed this session, let us know, reach out. If you have future workshop ideas, let us know. And hopefully this was valuable to people.